Prophase 1. This phase typically occupies more than 90% of the time required for meiosis. Chromosomes begin to condense. The homologous chromosomes loosely pair along their lengths, precisely aligned gene by gene. In crossing over the DNA, molecules and non-sister chromatids break at corresponding places and then rejoin to the other's DNA. In synapses, a protein structure called the synaptum. I just blind. How do you pronounce that?
Synaptic?
Synaptonomimal?
Yeah, sure.
Synaptonomimal? I don't know. I'll just reset. Alright, where was that?
Just say.
Can you edit all this out?
I can edit
Leave that
out.
all in there.
I really will leave it in there. I'm not editing that out.
Synapta. In synapsis, a protein structure called the synaptonomial complex forms between homologous, holding them tightly together along their lengths. The synaptonomial, which is not how you pronounce it, complex, disassembles in late prophase, and each chromosome pair becomes visible in the microscope as a tetrad, a group of 4 chromatids. Each tetrad has one or more schismata, crisscross regions where crossing over has occurred. These hold homologous together until anaphase 1. The movement of centrosomes, formation of spindle microtubules, breakdown of the nuclear envelope, and dispersal of nuclei occur as in mitosis. Can't talk. In late prophase 1, the kinetic chores of each
What are you looking at?
Look.
Homologue. I don't know.
The homologi. The kinetic chores.
Kinetic core.
Kinetic cores. The kinetic chores. In late prophase 1, the kinetic cores of each homologi attach to microtubules from one pole or the other. The homologous pairs then move toward the metaphase plate.
Meiosis 2. Alright. Prophase 2. A spindle apparatus forms. In late prophase 2, chromosomes, each still composed of 2 chromatids, move toward the metaphase 2 plate. Metaphase 2. The chromosomes are positioned on the metaphase plate as in mitosis. Because of crossing over in meiosis 1, the 2 sister chromatids of each chromosome are not genetically identical. The kinetic cores of sister chromatids are attached to the microtubules extending from opposite poles. Anaphase 2. The centromeres of each chromosome finally separate and the sister chromatids come apart. The sister chromatids of each chromosome now move as 2 individual chromosomes toward opposite poles. Telophase 2 and
Cytokinesis.
nuclei form. The chromosomes begin decondensing and
Cytokinesis.
occurs. The meiotic division of one parent cell produces 4 daughter cells, each with a haploid set of unreplicated chromosomes. Each of the 4 daughter cells is genetically distinct from the other daughter cells and from the parent cell.